Hi, everyone. Yeah, so I'll be talking about a project that finished last year on trying to generate a new type of probiotic for the industry. I'll start by just going through a bit of background, why we're interested in probiotics and what the idea was behind this. And then I'll have just a couple of slides, I guess, to explain 3 years of results and then some conclusions at the end. What was our original interest in really getting into probiotics? And it was really, with the withdrawal of antimicrobial growth promoters, there's a need for us to have other ways to try and maintain the health and increase the productivity of broilers, the way that the antimicrobial growth promoters helped us in the past. There are many sorts of products that can be used to try and maintain the health of broilers and increase their productivity. And I've got a list of them here. And we were particularly interested in vaccines and probiotics, but the project I'm talking about today is specifically on probiotics. But there are many other types of products that are being used to maintain the health of broilers. Probiotics specifically, though, the definition, the sort of official definition of these are that they're live organisms which, when administered in adequate amounts, confer a health benefit on the host. And that health benefit can be sort of wide and varied. A list of the ones that are sort of most interest to us in broilers are on this slide here. So there are potentially many different mechanisms by which probiotics can actually function. The overall aim is to improve the gut health of the broilers. They can do that by improving gut integrity. And of course, Reza talked quite a bit about gut integrity in the context of his low protein diets. A really important factor that the microbiota and by extension probiotics can have on birds is to stimulate their immune system and have it functioning properly. Some probiotics directly kill pathogens of interest to us, things like Clostridium perfringens. They can also stop colonization of pathogens and food poisoning organisms like Campylobacter if they sort of occupy a particular niche within the gut. And they can improve the overall gut microbiota. They can increase the production of short-chain fatty acids. They can produce enzymes that are important for nutrition scavenging. And more recently, we've even found that they can have an effect on the brain. They can affect behavior. There's claims that probiotics can actually improve the flavor of meat even. So there's a lot of potential reasons that we might want to use probiotics. But there are already an awful lot of probiotics on the market. Do we really need any more? And my answer to that is, well, the current ones, there are a few good products out there, I believe, but there are others that are perhaps less robust in their performance. There's quite a few products out there for which there's virtually no scientific peer-reviewed publications available as to what they'll do and to support the claims that are made. That's not true of all. There's good data for others. But a number of the products have sort of quite variable performance. They may work in one flock and they won't work in the next one. But with all of them that are currently available at the moment, there's a need for continuous dosing. And that need for continuous dosing tells us immediately that the probiotic isn't getting into the gut and staying there and sort of working as a harmonious part of the gut. And that's really one of the main issues that we aim to address in our project. We really wanted to get away from the need for continuous dosing, and I'll go on to sort of the outline of that in a second. But to try and explain our thinking of why we thought some of the existing probiotics had these issues, There are a number of reasons. A lot of the products that are out there, the probiotics that are sold into the market, it's the one particular bacteria that's sold into the different segments of the animal production market, so pigs, chickens, and cattle. And they have this sort of one size fits all. If it works in chickens, it might work in pigs and vice versa. And I don't know that that's necessarily true. So a lot of the probiotics that are actually sold into the chicken market don't actually derive from chickens. Some of the products out there have sort of poor viability. They're not quite what you think you're getting when you buy them. And in a lot of cases, the way that these strains have been identified is by using a whole series of laboratory tests to try and mimic some aspect of the gut environment. And it turns out that a lot of those tests that have been used very widely for decades haven't been well proven as being of real value. And we thought there were better ways to try and validate strains. But even with all these issues, if these weren't a problem, the fact is that the birds themselves have quite variable gut microbiota. So the actual environment within the gut is quite variable. Sorry about that. And you're asking the probiotics to work in quite different environments because each flock has a different background gut microbiota. So these are the sort of issues that we aim to address. So you have to think about the microbiota that's resident in the gut, and that microbiota has lots of different functions. And I won't go through them all. I haven't got time to do that here, but they're just listed on this slide, and you can see the sort of things that the resident gut microbiota are involved in. And by extension, probiotics that you put in potentially can affect those functions as well. So whether it's mucin degradation, short-chain fatty acid production, exclusion of pathogens, they can all be affected by the probiotics. So the key issues that we had aimed to address in our project was to overcome the variability in the gut environment and try and overcome the fact that all the strains that are currently used as probiotics have to be dosed all the time, so they don't persist. So this lack of strain persistence we thought was an issue that we could be addressed by selecting strains in a different way. So our goal was to first find bacterial strains that could reliably colonize, because we believed if they could reliably colonize under all conditions, then we would get reliable performance from them. And that meant that they had to be able to colonize in the face of that variable microbiota that I talked about as the background that's present in different flocks of birds. If we can get that reliable colonization in the face of the variable microbiota, it would mean that we'd have probiotic products that we'd only need to dose perhaps a single time at hatch, or only a limited number of times anyway. We wouldn't have to give continuous dosing. But the reliability was the big thing that we thought we could address. But this required the identification and development of bacterial isolates with very special properties. To find these bacteria, these potential probiotics with these special properties, we sourced the bacteria only from chickens. We went into high-performance flocks, isolated bacteria from there, and we abandoned some of those laboratory screening methods that I mentioned really haven't been proven, and we were doubtful as to how valuable they were. And we did all our screening, therefore, actually in chickens. So rather than on the lab bench trying to sort of mimic some aspect of the chickens, we actually screened in chickens. But the important thing to know about this project going into it, we'd done some work with lots of bacteria from chickens before, and sort of quite counterintuitively, we found that most of the bacteria that we pulled out of chickens couldn't reliably recolonize. That's because they come from a very specific collection of microbiota. So they come from a very specific environment. They depend on all the other bacteria that are in there. And so they can't always go back into any other old chicken that has a different sort of microbiota background. So we already knew going into this that typically lactobacillus bacteria and E. coli, of the ones we pulled out of chickens, only about 3 to 5% could reliably go back in and persist in newly infected birds. So that's what we were up against. We were needing to screen lots of bacteria to find these special ones that could persist. So this is the method that we use. So this is what we ran during the project. So we went into broiler flocks, isolated a whole heap of different bacterial strains. So these grey lines up here are supposed to represent uncharacterized bacteria. We then went on and used a couple of different methods to actually characterize those bacteria. So we knew exactly what they were, what species they were. We could identify them. And a lot of those bacteria were of the same species and they are difficult to tell apart, but there are also lots of different species that we were using there. And we could differentiate them relatively easily. And what we did was to take bacterial species, bacterial strains that we could differentiate, and we pooled them into this ordered array of colored bacterial cartoons down the bottom there. We made a pool of those bacteria, so lots of different bacterial species within that pool, and we used that pool to re-inoculate birds. And because we could tell all those bacteria apart, we could then work out what we could re-isolate from those birds. So we put in pools of about 10 to 15 different bacteria, and typically we'd pull out 2 or 3 of those from the end. And these would be our candidate probiotic bacteria. So that's the screening process. And we needed to be able to do it in this way because we knew we had to be able to screen hundreds of bacterial strains to find the few that had these special properties that we were interested in. Now, we did all these experiments in the chicken isolator facility at the University of New England, and Priscilla was invaluable in helping us do this and giving us access to these facilities. So in each of the experiments we did in this facility, we could test up to 24 different groups of bacteria. So I said each group of bacteria had about 10 to 15 different bacteria in. So we could screen about 200 different bacteria in one experiment. Now, previously, when people had done this sort of work, they typically used 1 or 2, maybe if you're lucky, 3 or 4 bacteria to test whether they had probiotic properties or potential. By using this new method that we developed, we were able to screen hundreds of bacteria. So what we did in the end, we're back to this cartoon here. We isolated thousands of these sort of uncharacterized bacteria up here at the top. Then we characterized and we got good characterization, identification of 797 different lactic acid bacteria of 31 different species. And of those characterized isolates, we put 210 unique isolates back into birds. And of those thousands of bacteria we started with, we've ended up with a collection of 50 candidate probiotic bacteria. So these are bacteria that persist in birds. So we can put them, we've pulled them out of chickens, we can put them back into chickens, and they reliably recolonize in those birds. But the fact that they can persist and recolonize doesn't, at this stage, make them probiotic bacteria. We have to do a lot more to show that they're probiotic, that they actually give some advantage to the host. So a few of the things that we've had a chance to look at are whether any of these bacteria have antimicrobial activity. And yes, some of our candidate bacteria do. So some of them kill this food poisoning bacteria, Listeria. Perhaps more relevant to us at the moment is that some of our candidate probiotic bacterial strains actually kill Clostridium perfringens, the organism that's involved in necrotic enteritis. So that's a potentially beneficial thing for these strains to have. The other thing that we found when we were screening the bacteria in these small groups of birds, we were taking bird weight. So these trials weren't really designed to look at productivity, but even with the very small number of birds that we had, we found that we did have differences in the average weights in different groups. So the groups along the bottom, they're just different pools of bacteria that we've used. And you can see that there are differences in the bird weights that are coming out of these groups. So that's potentially interesting too, that some of these bacteria could be having positive effects on the weight of birds. There are also differences in the actual weight of the gut and the length of the gut in some of these birds. So unfortunately, this project was going on right in the middle of COVID, so we were really severely restricted with the trials that we could do. We were lucky to get through that initial screening phase, but we haven't done enough work on the actual probiotic properties of these bacteria, so that's what needs to be done now. So these are the trials that we'd like to be able to go into now, although the project funding has finished at the moment. So we want to screen those special persistent isolates that we've got through all that screening process to look for improvements in gut integrity, improvements in immune system, pathogen carriage, and importantly, the productivity. And we think that by using the right collection of these probiotic strains and inoculating birds very early on, we can improve the uniformity of flocks. So if we can find the resources to do these, we believe that in the end, we're going to have some unique new probiotics that will be ready for commercialization and industry use, but we're not there yet, unfortunately. So the outcomes of this project, we've got probiotics, next generation probiotics that are tailored for chickens. They can be reliably delivered to flocks and persist in those flocks. We've got some evidence that they could support health and productivity, but there's a lot more that we have to do there. All the work we've been doing up to now has used just a single dose of these, and we've been able to get them to take in the microbiota of the birds and persist. And we believe that there'll be a lot of cost savings because of the limited need for dosing and hopefully the reliable efficacy of these things. So just to finish up, of course, I'd like to thank AgriFutures for the support. This was a big project. Had a number of people within the RMIT lab who are heavily involved in it. Hal Van, Josh Fraser, me and Chigor, and Priscilla at UNE. She's in Hong Kong now. Access to the facility to actually do the trials was invaluable. And of course, the industry vets who got me access to the healthy flocks from which the bacteria originally isolated. So, okay, thanks. That's it.